All right, so for this week, we're going to focus on female reproduction in lecture. And let's begin with some very basic anatomy. The ovary is the gonad of the female reproductive system. It produces sex hormones differentially to the testis in the male. It also produces eggs, which are the functional follicle units of the female reproductive system. The eggs develop into fluid or within filled sacs called ovarian follicles. Just like follicle stimulating hormone can lead to the production of sperm in the male, it would lead to egg development within the ovarian follicle in the female. And once fully developed, the egg would be transported into the uterus through the fallopian tubes. It is during this transportation phase that fertilization would occur within the fallopian tubes. The uterus is primarily the site for fetal development. Focusing on the ovary here in a little more detail, it is important to note that it would contain multiple follicles at different stages of development. In fact, the woman would be born, or the birthing person would be born with all the follicles that they will have at birth, and then eventually these are used through cycles until menopause, where the ovary begins to, well, essentially fail. In one egg cycle, the primary follicle develops into a primary oocyte, which is ultimately released for transport to the fallopian tubes. It is important to note that the tissue surrounding where the oocyte was plays an important role in endocrine signaling as well. This tissue becomes known as the corpus luteum, which means yellow body in Latin. The corpus luteum is especially worth noting and something we will come back to because it plays an important role in regulating both the menstrual cycle in primates and the estrous cycle in rodents, dogs, etc. It is likely that many of its hormonal functions were selected for because they promote a healthy place for fetal growth, but the progesterone that it produces has many behavioral and physiological effects that we'll talk about by the end of the lecture. Now let's switch to a broad overview of the female HPG axis, or hypothalamic pituitary gonadal axis. There are some similarities to what we just studied in males, but also some important distinctions as well. As should now be familiar to you, the gonadotropin releasing hormone would be released actually from the hypothalamus and then act on the anterior pituitary, which would then in turn release luteinizing hormone and follicle stimulating hormone. These act on the ovaries to cause the release of estrogen, progesterone, and inhibin. One big difference between the HPG axis in males and females is how feedback regulation occurs. In males, you have a much more simpler form of negative feedback, which is, more sort of the stereotypical example of what we've studied really from the first week of class, a simple negative feedback. You have too much of the chemical being produced, it'll automatically shut off once it gets too high, like a thermostat. In females, the ovarian hormones act at both the pituitary and the hypothalamus, and their effects are dependent on their overall levels. In other words, sometimes you get negative feedback, and sometimes you get positive feedback to further enhance hormone secretion. Estradiol is one of the primary ovarian hormones we're going to consider in this lecture. It is synthesized by granulosa cells in the ovaries, and its synthesis is triggered by follicle stimulating hormone and luteinizing hormone. Estradiol acts on the central nervous system to maintain libido and sexual behavior. It also provides feedback regulation of gonadotropin releasing hormone, luteinizing hormone, and follicle stimulating hormone. Although it isn't always unidirectional negative feedback as occurs in the male. Things do get complicated depending on overall hormonal levels. Uh, Estradiol also plays a critical role in egg maturation, lactation, giving birth, weight gain, and bone mineral deposition. We will also talk a lot about progesterone in this class. Progesterone is mainly synthesized by those squishy yellow cells that we just went over called the corpus luteum. Progesterone synthesis is triggered by luteinizing hormones, but its synthesis is primed by follicle stimulating hormone. FSH, or follicle stimulating hormone, must be present to set the stage or prepare the environment for luteinizing hormone, or LH, to trigger the synthesis of progesterone. Progesterone acts on the CNS to increase sexual receptivity. It has such a pronounced effect on metabolic functions that your body temperature can increase or change by as much as 1 degree following a progesterone shift. Progesterone levels are high during pregnancy and contribute to things that you hear birthing people report, like hot flashes. Reproductive cycles are most often thought of in the context of female reproduction. These are most commonly associated with shifts in the hypothalamic pituitary gonadal axis. There can be seasonal shifts that are very pronounced in some species. For example, light or even temperature can trigger shifts in the HPG axis that translate into changes in sexual behavior. In Colorado, even here in the mountains, you think of seasonal shifts in sexual behavior, going to watch the elks bugle at each other in these different groups of male and females. It's really a beautiful, amazing thing to see if you haven't. I highly recommend it. But the menstrual cycle in primates is a little different. It's not as sensitive to environmental changes as the estrous cycle that occurs in most other female mammals. And I do think it is important to reflect on the differences here and really throughout the class between the menstrual cycle and the estrous cycle, especially because having done it, I find studying the estrous cycle to be difficult, uh, especially when you take into account that things like the sex of the investigator, whether or not we are exposing the animal to drugs. For example, I tend to be a drug researcher. So if we give an animal a lot of methamphetamine, their cycle can instantly shift or stop. Similarly, that can vary depending, especially if I'm doing it as a male. And then if I'm stressful at doing this, this can also shut down or shift the estrous cycle within a matter of minutes. Thus, I do believe that there are estrous cycle experiments in rodents that we should be considering the validity of when we're making claims about sex hormones in primates. It's just something to keep in mind. And I also find it odd that I cannot have an animal self-administer methamphetamine without seeing massive changes in the estrous cycle. But other labs that are sort of writing the books on this seem to have no real issues. And I don't know exactly what's going on there. It's possible that in the novice, like me, that isn't a sex hormones, haven't studied sex hormones experimentally my whole life. Maybe that's part of the problem. Maybe that should be for experts only. But we've also changed NIH rules, so everybody has to consider sex as a variable in their studies. So yeah, I don't know. I don't know exactly what's happening there, but it is something that it's definitely caught my attention. But in general, in either the estrus or the menstrual cycle, you should be able to detect a visible sign of ovulation, and this visible sign is thought to have been selected for to ensure the female knows that this sort of mating cycle has ended. When we think of the human menstrual cycle, things are much more consistent than the ester cycle, where things can change very rapidly and be influenced by environmental variables very readily. And the menstrual cycle also allows us to count days. In the rat, the cycle would be over the course of days, with just a few days, 4 days, or hours even, rather than a standard 30-day cycle. And of course, there is variance here. Probably 21 to 35 days would be the more the confidence intervals there, around a 30-day average. But whether or not you're 21 days, 35 days, or 30 days, there would be some sort of more consistent pattern that wouldn't change with the weather, quite like you see in other species. In primates, typically the cycle does occur monthly, and menstru literally means month in Latin. Menstruation is used to indicate the periodic shedding of the endometrium, which becomes thickened prior to menstruation under stimulation by ovarian steroid hormones. The thickening involves nutrients being added to promote successful fertilization in addition to increased vascularization. The shedding of the endometrium is accompanied by bleeding and can be thought of as day 0 in the menstrual cycle. This figure is admittedly complicated, but it's also very important and summarizes so many important concurrent concepts that are occurring here that I really want to break this figure down in detail more than pretty much any other we're going to study in this lecture. So first, let's break the menstrual cycle into 3 phases. The first, you can see here to the left, would be the follicular phase. The second would be ovulation, that sort of bolder dashed vertical line. And then you see the beginning of the luteal phase. And I guess you could throw in a 4th period of menses here if you wanted to. But during the follicular phase, let's break down exactly what's happening. So during the follicular phase, estradiol rises over time while most other hormones and body temperature remain relatively stable. You do see a somewhat important slight increase in follicle stimulating hormone, but it pales in comparison to that drastic rise that you see in estradiol. You can also see that the body temperature also remains relatively stable during this phase. And you can also see that the follicle up at the very top there is growing within the ovary during this phase. Then at ovulation, you see a dramatic drop in estradiol that is followed by a spike in luteinizing hormone and follicle stimulating hormone. You also see a clear body temperature jump, but really as much as 1 degree in body temperature here. Then you see the onset of the luteal phase. During the luteal phase, progesterone levels become most pronounced. The progesterone is primarily being released from the corpus luteum. LH and FSH are back on the floor, back to low level, but body temperature remains elevated until progesterone begins to drop at the end of the luteal phase. During this phase, nutrients and increased vascularization are being added to the uterine wall. If the egg does not implant into the uterine wall, menstruation occurs. So here I want to briefly touch on menopause, and then we'll get back into briefly what happens with fertilization or non-fertilization. But menopause is the cessation of ovarian activity. And there are some similarities to andropause, which we studied last week during male reproduction. However, there are also some important differences. unlike in andropause, where there are three main causes that lead to most men experiencing it, and by most, really not all. In women, all females experience menopause if they have ovaries that were producing ovarian hormones. And then the loss of the ovaries would lead to essentially estrogen withdrawal, which is menopause. Menopause begins due to what could be called a failure in the ovary, not the pituitary. This isn't a brain deterioration thing. The ovaries, and fail is sort of a harsh word. What's really happening is the ovaries become depleted of follicles and stop secreting estradiol. Recall again that women are born with all the follicles they will ever have, and each is selected during one cycle until all have been depleted. It is also actually pretty interesting to think about what causes a follicle to be picked at a certain point in time. I think there's a lot to be um, figured out in that regard. But once there are no follicles, the ovaries begin to fail at their primary function, which is to make and secrete the follicles and the eggs. So this is really all due to gonadal aging, the ovary aging and using up all the follicles, not the brain. Even when the ovaries stop producing estrogen, there is still some being released into the blood from the adrenal glands and adipose tissue if there's a large buildup of adiposity. However, these levels are much lower, and most behavioral and physiological symptoms of menopause are thought to result from withdrawal from the estrogen levels itself. So now I want to get back into the egg itself here and what happens to hormones in the case of fertilization occurring. So we've already gone over a little bit about the cycle and what would happen at the end of the cycle. Well, the new cycle would begin. We would switch from luteal to follicular after the shedding of the menses. However, what happens if the sperm successfully fertilizes the egg? One unique thing that happens is that the corpus luteum keeps producing progesterone to maintain the pregnancy. This is actually very relevant to know from a biomedical and treatment perspective. In the medical setting, medical professionals will actually prescribe progesterone if the birthing person reports experiencing repeated miscarriages. The idea here is that progesterone is important to maintain the pregnancy. As a result of heightened progesterone feedback, you tend to see a decrease or a complete inhibition of luteinizing hormone and follicle stimulating hormone. That, in turn, turns off the menstrual cycle. So you would stop having the monthly period. If fertilization doesn't happen, you would get exactly what we've already studied on that chart. You should see the scenario where progesterone levels should go back down as the corpus luteum breaks down. The uterine lining would also break down, leading to menstruation. And then follicle stimulating hormone and luteinizing hormone would begin producing again to initiate the next full cycle, which again occurs usually between 21 to 35 days, with an average of really 28 to 30 days. One hormone that is produced only in pregnancy is called human chorionic gonadotropin, which rescues the corpus luteum. When the embryo implants HCG, or human chorionic gonadotropin, begins being released very rapidly and at very high concentration. and helps set the stage for a successful pregnancy. Early indicator pregnancy tests are looking for this very chemical. They measure levels of HCG, which should double every 2 to 3 days for the first month of pregnancy. Now, let's talk about how these cycles, and we'll start with the estrous cycle here, is associated with female sexual behavior. First, I want to reemphasize that important distinctions between the estrous cycle and the menstrual cycle exist. Again, the estrous cycle only lasts for about 4 to 5 days and can be greatly influenced by environmental variables. The menstrual cycle is much more consistent and doesn't influence sexual behavior as drastically. Sexual behavior in rats is most often studied in the lab because it's easier, cheaper, and safer than doing the same using primates. Lordosis is the curvature of the spine in a sexual position females will observe that physically allows the penis to enter the vagina. If the female doesn't assume this position, it's very difficult to make the penis fit in the rat. Females can be observed as aggressing upon or even injuring male rats who persist in their sexual advances despite them not being in estrus or exhibiting lordosis. Primates are different. First, sexual activity is not limited to ovulation, and different species will show unique sexual behaviors. One theory to explain this is that in many mammalian species, sexual behavior is directly tied to ovulation and reproduction due to simple natural selection. But in the primate, we have much more complicated social interactions and societies. Thus, sexual behavior may have evolved so that it is beneficial to help one thrive in many ways, not just to reproduce. And if one is doing better socially and sexually, it is also much more likely they will reproduce in our more complex societies with an ideal mate. We've already brought up chimpanzees and bonobos, but I think it's worth considering these two distinct species again. But both species are about 99.9% genetically similar to us. Chimpanzees seem to use sex not only to reproduce, but also to exert dominance and maintain social hierarchies. Bonobos seem to use sex more for conflict resolution and also engage in much more homosexual behavior. Again, chimpanzees tend to be more male-dominant societies. Bonobos tend to be more female-dominant societies. Interestingly, both males and female bonobos engage in homosexual activity, whereas male chimpanzees are much more likely to do so than female chimpanzees. And when the chimpanzees do engage in the homosexual behavior, it seems to be directly related more to social dominance than conflict resolution, pleasure, just sharing the fruit. I will also point out that chimps develop more sophisticated tools and are much better at war. There are some interesting differences between these two species. My biggest point, however, is that primate sexual behavior tends to be more complicated than we observe in rats, and there is a lot of variability out there, not only in humans, but in primates. Sex isn't just about reproduction and isn't limited to only occurring at times of ovulation. Now I'd like to go over briefly the components of female sexual behavior. When we covered this in males, I broke things down into appetitive and consumatory behavior, with appetitive behavior being the game or the hunt, the going out and looking for a sexually receptive female, and then consumption. While there can be considerable variability here, it is generally more reflexive. It tends to be somewhat surprisingly what males put a lot less effort into versus sort of the game or the hunt. It also brings out different areas of the brain. You see sort of similar activation during the pursuit of sex as you would during the seeking of a drug or the seeking of food, whereas the parts of the brain that are active during copulation for the male are going to be much more similar to eating, a good dinner, or it's a little different. It engages the brain differently. So we typically break things down to three components of female sexual behavior: attractivity, the stimulus value of a female for a given male. This is mainly a hypothetical construct and inferred by observation and measured in relational terms. Proceptivity, the extent to which a female initiates copulation. And receptivity, the state of responsiveness to the sexual initiation of another individual. This, as you can imagine, is very difficult to perfectly measure in a rodent, even in a non-human primate, without vocalization of what one is experiencing. We tend to measure this just in terms of postures observed. We've already learned through our reading this last week that these categories can interact and overlap with hormones in real-world examples. One example that was discussed was the sort of the stripper study, in which it was demonstrated that during the follicular phase, or when a woman is closer to ovulation, they can observe higher levels of tips earned, particularly if they are not on the hormone regulating pill. So this would be, I think, clear example of the interaction of attraction or attractivity and hormones. Here is another example here. These are AI generated images of two male faces, A and B. And it turns out that whether or not you're in the follicular or luteal phase can determine who you rate as being more attractive. In the luteal phase, with high levels of progesterone, people tend to rate person B as being more attractive. You would see the same in surveys of pregnant women or pregnant people with high progesterone levels. Follicular phase is most associated with preference in person A. And, the same person, you could even just do this longitudinally and see the same person rate person A as being more attractive up into the point of ovulation. And at that point, person B here, would be a more desirable mate. Attractivity here would be measured typically in choice experiments, like you can see illustrated here in the rodent, where you would have two potential sexual mates on two different sides, and you measure which one the other sexual conspecific seeks out. You tend to see markings and swellings emerge during periods of around ovulation or a peak sexual receptivity. The certain monkey species in particular tend to show large red swellings, around the pubic area, which is literally to, flash a light at the male saying, "Look over here." Estrogen tends to enhance attractivity. First, it's worth noting that if you remove the ovaries from a female animal, something known as an ovariectomy, typically abbreviated OVX, the OVX females without their ovaries are rarely mounted. This is something that can be reversed with estrogen therapy, or if you inject the animal with estradiol after the ovaries have been removed, you can see mounting resume to a almost normal level. Proceptivity is another one that we should talk about. All appetitive activity shown by females, and these tend to be socially solicitous behaviors. It's also worth noting that this is just displayed more typically in female behavior. Around 80% of all proximate social interactions between male and female monkeys is actually initiated by the female prior to mating. And estradiol enhances proceptivity in the absence of male interest. If you have increased estradiol levels, this will increase, proceptivity regardless of whether or not there is a receptive male. Another important thing to keep in mind when we are thinking of menopause, just as another fun story here, this has happened to me just the other day at the coffee shop. I was waiting in line and chatting with the barista who pointed out to me, I mean, like half of her job, other than making coffees, is talking to everybody that comes through this line. And she noted that the men in line in particular, uh, you know, she finds herself an initiator of not only conversation with the different men, but in their interactions with each other. I think her exact words were, "If it weren't for me, all you men in line would only ever say yup to each other and nod your head and go on your days without ever even having a conversation with each other." And it really, I think, is somewhat this in a nutshell. That yes, like she was through proceptivity in a way, facilitating conversation, really at a large social level. And a lot of that depends on estradiol. Then we have receptivity, the consumatory phase of mating behavior. This does involve some species-specific mating postures, a little harder to study in animals, but we do so by measuring changes in postures or posturing of the animal itself. You also see engaging in sexual activity and cuddling, increasing testosterone in women, which is thought to feed back and increase receptivity further. And estrogen, of course, stimulation is very important for receptivity. Overectomized females do not display mating postures. You must have the ovaries in place. One interesting finding, this is from a study on involving rhesus macaque monkeys, which are smaller monkeys, definitely further down the evolutionary scale or axis or tree than the chimpanzee or the bonobo, which are going to be much more, Homo sapien-like. Still, this is an interesting study from rhesus macaques that demonstrate how social factors in estrogen can interact to control sexual receptivity. So in this study, they investigated the effects of a male and a female being placed together in either a small testing environment. Think, more like a private room with just the male and the female present, whereas the other would be a large colony, put together, composing many different monkeys from the troop. And then they studied the consequences of the male-female interaction and were particularly interested into whether or not male ejaculation occurred during the exchange or the interaction. So what they found was that if the male and the female monkey interact as a pair in the small private area, about 50% of the interactions ended with ejaculation, irrespective of what day it was in the female cycle. However, if you look around peak ovulation, you find that in the social colony setting, you only saw about 50% of the interactions turn into an ejaculation outcome around this peak ovulation. So, one way of looking at this is maybe it's sort of like the endocrine power of ovulation overcome social factors. It might otherwise reduce or attenuate sexual behavior here. And it's just like the ovulation power is so strong that you're going to go through the sexual behavior irrespective of what the ant next that's sitting next to you might be thinking about it. Whereas the social influence is still there a little bit. When you're looking about the outcome of the ejaculation in the private area, but much to a much less noticeable degree. In human diary studies, it's also been shown that women think about sex more around ovulation and are much more likely to initiate sex. However, I do think it's important to note that they do not appear necessarily more receptive to male advances. If anything, it still seems to involve the same level of selectivity, just you're more interested in the mate that you might otherwise be interested in. It's also worth mentioning testosterone here, which definitely deserves to be discussed in the context of female sexual behavior. Recall, and I've said this many times now, that even though we tend to think of estrogen as the female sex hormone and testosterone or androgen as the male sex hormone, both are expressed in males and females at different levels. And it turns out that testosterone is probably very important in mediating female sexual behavior. First, blood sample studies in humans have revealed that testosterone levels are lower in monogamous women than in those who are involved with multiple partners. We also see that testosterone levels are higher in new relationships, but these levels decrease with time. And we've already discussed that cuddling and intimacy after sex can increase testosterone, which can in turn lead to more sexual behavior with a new partner or possibly even other partners. And in general, low testosterone is associated with low sexual motivation, which is especially important within the context of the pill. Certain types of birth control, like the pill, are known to reduce testosterone levels, which can also reduce sexual behavior. So this is something I wanted to go over, the pacing of copulation, and I first want to point out that this hasn't been studied very well in primates. Most of what we know about this phenomenon is from rodents. But in rodents, we know that, or have known for some time, that the pacing of sexual behavior can enhance reproductive success. The idea here is that the pacing of the entries, not really like depth or anything like this, but the pacing maintains luteal function so that the fertilized egg can be successfully implanted. It's believed and what's been shown that a steady rhythm seems to promote an ideal environment for implantation. So I guess one could say there maybe is something to this motion of the ocean thing. And it's also interesting to me to consider how important dancing and rhythm seem to be in mate selection. And maybe there is a very deep evolutionary reason for that. So now I just want to briefly go over pheromones here. I'm not going to hold you to every little detail here. Again, stick to your study guide. But a lot of what we know from pheromones, there seems to be some species-specific important effects of pheromones. And while there are certain effects in humans that we are going to go over, they might play a less pronounced role than you see in other species, but still an important role. They certainly produce effects. But what do the data show? So first, we're going to talk about pheromones in rodents. One pheromone that we're going to talk about comes from females, and the other, and this is thought to suppress both a luteinizing hormone and follicle still stimulating hormone from the pituitary. The other is emitted from males and can accelerate puberty, induce estrus. So estrus, when it's spelled like this, estrus, tends to be the estrus phase of the female animal. Estrous is the estrus cycle. So estrus with just the U would be just one phase of the estrus cycle. So, but anyway, this pheromone is known to induce estrus in females when it's coming from males. So that would be sort of induces sexual receptivity. It's also been shown to interrupt pregnancy depending on the reproductive condition of the female. This also induces an abrupt release of luteinizing hormone, which stimulates follicular growth. There are several pheromone-mediated effects that have been observed primarily with rodents in the laboratory setting. One is known as the Lee-Boot effect. When housed in groups with no males, female mice display longer estrus cycles. Another is the Whitten effect. Here, if you have in the presence of a male or his odors, this can induce estrus behavior within 48 hours in group housed female mice by inducing gonadotropin releasing hormone. Exposure to female urine suppresses gonadotropin releasing hormone and estrus. The Bruce effect is very important and thought a lot in the animal research space because it is what impacts the breeding of the animals the most. But here, pregnant females abort or resorb fetuses if exposed to male or male urine that was not the sire for 48 hours by inducing gonadotropin releasing hormone release, which is incompatible with pregnancy. So here it would be very important that if you are responsible for putting the males and females together for breeding purposes, that you would keep them isolated and then not have much contact with them if you were working with a lot of other male animals, which tends to be the case. Now that you also have the Vanderberg effect, which it's been shown that female juvenile mice exposed to adult males or their urine mature earlier than those not exposed by inducing gonadotropin releasing hormone. Exposure to adult females or female urine inhibits gonadotropin releasing hormone release and puberty. One important study in humans that really documented or really, I think, proved a lot that pheromones can play an important role in humans as well came from Wellesley College. So in Wellesley College, at the time, it was primarily a women's college in Massachusetts, where here women lived together, of course, for extended periods of time, and were shown to eventually begin synchronizing their menstrual cycles. It was also found that this required about 7 to 8 months of cohabitation. Martha McClintock, a senior at Wellesley, convinced all 135 of her dorm mates to keep track of menstrual cycles for a full academic year. And she eventually published her work in Nature, arguably the most important and prolific scientific publication that exists. It's pretty much unheard of for an undergraduate student to put a study together like this together and publish in such a prestigious journal. It's also worth noting that Hillary Clinton was a senior at Wellesley at this time, and there are only so many dorms, and she's never confirmed nor denied if she was one of these 135 people, but it's widely speculated. So the existence of an importance of human pheromones was pretty much unproven until 1998, and if anything, there was quite a bit of skepticism about it, and really there's still some skepticism about the power of pheromones in people versus other animals. But as this study shows, I mean, there's clearly an effect. So McClintock had women provide chemosensory samples during different times of their menstrual cycle by wearing absorbent pads under their arms for at least 8 hours a day. Pads from the follicular phase accelerated the peri-ovulatory surge of luteinizing hormone and shortened menstrual cycles of women exposed to pads for 1 to 14 days over 2 menstrual cycles. Pads from ovulation delayed the luteinizing hormone surge and lengthened the menstrual cycle of recipient women by 1 to 12 days. There's additional support for pheromones and their effects in humans. One is smelling, a derivative of anabolic steroids, can activate the hypothalamus to stimulate the release of gonadotropin-releasing hormone in heterosexual women and heterosexual men. And I think this is very interesting and possibly quite important, but not in homosexual men and heterosexual men. So it really, rather than whether or not you're XX or XY, it's whether or not you're attracted to men. It doesn't matter if you're male or female here. Yeah, but there only seems to be the effect of the steroids on the hypothalamus here if you're sexually attracted to other men. I also want to point out that illness suppresses female sexual behavior, but not males. Females with low energy or poor conditioning are going to have less attractivity, less receptivity, and less proceptivity. Here in one study, they activated the immune system by cytokines and showed that this inhibits gonadotropin releasing hormone gene expression, decreases circulating luteinizing hormone, and inhibits steroid hormone synthesis in the gonads.